Go ahead and introduce our speaker today, Li-Ming Tai, who, as you know, just begun a month ago a Stengl-Wyer postdoctoral fellowship. Li-Ming came from us originally from Harvard University, where she got her PhD in 2000, working with Chuck Davis on angiosperm family, the Malpighiaceae. And she then did a postdoc in 2021 in the Department of Botany and Plant Sciences at the University of California at Riverside. Li-Ming is actually jointly working with myself and Justin on a group of parasitic plants in the Orbancaceae. Li-Ming's credentials are excellent. She's published 9 papers, including in high-profile journals such as Current Biology, Systematic Biology, and New Phytologist. Won a number of awards during the years. 2 in 2021, the W.D. Hamilton Award for Outstanding Presentation at the Society for the Study of Evolution, and the Dorothy Skinner Award also in 2021 at the Society of Integrative and Comparative Biology. 2 other awards she was finalist for in 2019 was the Ernst Mayr Award for the Society for Systematic Biology, and the Rising Star Award in 2019 in Organismal Botany from the Society of Integrative Biology and Comparative Biology. So it's a pleasure to introduce Li-Ming, and she's going to present a talk on anything but the species tree. Li-Ming.
Thank you, Bob, for the nice introduction. And thank you so much, everyone, for coming today. And I would like to thank the Department of Integrative Biology for having me today. So with that being said, let me share my screen and get it started. Can all of you see it? All right. Okay. Here we go. So anything but the species tree. Obviously, I am going to talk about phylogeny. Phylogeny, namely constructing and evaluating hypothesis of relationships. It really provides the most fundamental knowledge that we need to understand the evolution of any groups of organisms, including, for example, their trait evolution as well as biogeography. For example, here, by mapping traits to these phylogeny of plants, we can understand the importance of the evolution of cuticles and protected embryos, etc. And how does these traits help the plants to conquer the land. The concept of phylogeny can be applied to both species as well as to individual genes. So here, a species tree often refers to the evolutionary history of the organisms itself, while on the other hand, the gene trees often describe the evolution history of these individual gene trees. And these 2 trees, they do not necessarily overlap with each other. And then we call this phenomenon as gene tree species tree discordance. For example, here you can see on the lower right, there is a gene tree that actually differs from the topology of the species tree. And this could be due to a number of reasons. So I will begin today's talk by introducing you some of the conceptual backgrounds in species tree gene tree discordance. So here in the 1997, Wayne Madison wrote this conceptual paper, Gene Trees in Species Tree, in which he visualized the variations in gene tree as a cloud of gene histories. So this illustrated a really intricate relationship between gene trees and species trees due to the evolutionary forces that act on genes within and across the branches of the species tree phylogeny. And then when all of these add up to a genome level, you can get a really fuzzy distribution of gene tree variations, as exemplified here in the North American cacti. The blue gene trees are the gene trees that match the species tree topology. But on the other hand, you can also get these green and red gene trees that differ from the topology of the species tree. And then further investigate of these gene trees reveal that they are caused by these ancient hybridization or integration events that took place in the past. So the take-home message is that some of these gene tree variations, they contain a rich source of biology of the species. And then I will then introduce 3 major biological processes that can lead to the gene tree species tree discordance, as these concepts will come up repetitively in today's talk. So first, gene duplication and the subsequent biases in gene loss can lead to a difference between the gene tree topology and the species topology. And secondly, horizontal events, including both introgression as well as the horizontal gene transfers, can introduce reticulations in the species tree. And then this will cause variations in gene tree as well. And this is because different part of the genome will trace back to different parental lineages. And thirdly, incomplete lineage sorting, which might need a little bit more explanation. It happens because species does not trace back to a single breeding pair. And this phenomenon is especially prevalent when the population size is large. For example, if you track the red, the black, and the blue lineages in a plot, you can get a gene tree topology here that actually differs from the species tree. And then the overarching goal of my research is to use the biological information that embedded in these gene tree variation to study the plant genome evolution. For example, the duplication can tell us something about the genome duplication happened in the past. And then the reticulated phylogeny can tell us if there's any hybridization happened. And then finally, the incomplete lineage sorting can tell us something about the population size dynamics of the organisms. So, and then I would also want to investigate, of course, the environmental and the biological factors that drives such evolution. So the plant group I've been work in the past is called Malpighiales. It's a textbook classic clade of adaptive radiation that took place sometime in the mid-Cretaceous. In this group, you can find mangroves, temperate willows, passion fruit. It also includes some of the most bizarrely modified plants on the planet, including the endoparasitic Rafflesiaceae, and then the aquatic Podostomataceae, as well as the old wood cacti, in quotes. These are actually Euphorbiaceae species. So in Malpighiales, there are more than 16,000 species, which accounts for about 8% of the eudicot diversity. However, this group is also notorious for its recalcitrant relationship. On the right is a simplified phylogeny of this group. And as you can see here, that this rapid radiation at about 120 million years ago leads to really short internal branches in the backbone of this group, which leads to really recalcitrant relationships. And then within this short window of 8 million years, all of the family level diversity, including many of which we have just seen, has evolved. So to better illustrate the recalcitrant relationship, I would like to show you this somewhat reformatted phylogeny of angiosperm, the flowering plants. So here, the nodes and edges can be interpreted as the normal nodes and edges that we see in a normal phylogeny. However, we can see there are tangled branches here and there, and these tangled branches suggest where phylogenetic conflicts resides. And then here, this large messy ball is where Malpighiaeli sits. Moreover, across the entire plant tree of life, out of 10 most top unstable nodes, 9 of them are within Malpighiaeli. So I hope these give you an idea of how difficult it is to resolve the phylogeny of Malpighiaeli. And then today's talk, well, there will be 3 major sections, and then I will visit each of the 3 phenomena that I have just introduced that can cause gene tree variations. I will investigate how these 3 phenomena affect the recalcitrant relationship in Malpighiaeli, and also what does these variations can tell us in terms of the natural history of Malpighiaeli. Okay, first, whole genome duplication, or WGD for short. It has been increasingly identified across the tree of life in eukaryotes, but it is especially abundant in plants compared to animals or fungi. One important outcome of whole genome duplication or any kind of gene duplication is that it introduces novelty into the genome. So this hypothesis was originally proposed by Ono in the 70s, in which one of the duplicated copies is released from the selection pressure such that it can evolve novel functions. And this phenomenon is called neofunctionalization. It can generate variations in genetics, and also it can also generate morphological variations, which can be potentially adaptive in certain environments. Indeed, examples from wild strawberry has demonstrated that polyploids, which is shown here in red, they demonstrate higher fitness across 3 different heterogeneous environments compared to their diploid progenitors. And such adaptability can be especially important for plants living in harsh environments. And then one extreme example of these comes from the mass extinction events. So here is, again, another simplified phylogeny of flowering plants. And what we can see here is that these identified whole genome duplication, they are clustered around the KT boundary, which is in mass extinction events that is hypothesized to wipe out the dinosaur population. And then it is possible that the additional adaptability and flexibility to cope into new environments in these polyploid plants that help these plants to survive through such mass extinction events. And then in Malpighiales, WGDs are identified in heavily studied genomes at that time. These genomes represent economically important plants, including the rubbers, the castor beans, flax, etc. However, due to the limits in taxonomy sampling in these genome studies, the researchers is unsure about the precise placement and the timing of these events. So to more accurately characterize these events, we expanded our taxon sampling within Malpighiales by introducing around 30 newly generated transcriptomes that represents nearly all of the family level diversity. And here I want to acknowledge our collaborator, Sugu, in the University of Malaya for helping us with the collecting these plants. They all live in remote tropical areas. Sometimes they're really hard to get them. And then in order to place whole genome duplications onto the species tree, we use the method called gene tree species tree reconciliation. So this method tries to explain the differences between the species tree and the gene tree by invoking process like duplication, loss, and transfer. For example, in order to explain the gene tree here on the right, we need to invoke a duplication at the root of the phylogeny, as well as a loss in species B. So using this method, we mapped more than 5,000 gene trees to species tree, and then we identified a surprisingly abundant whole genome duplication events in Malpighiales. So these duplication events are marked by these orange circles in the plot, and then the size of the circle indicates the proportion of genes that support the duplication events. So the 24 whole genome duplication we identified here further adds to the growing consensus that whole genome duplication is a widespread common phenomenon for flowering plant evolution. What is more interesting is that these events, they are not randomly distributed across time. Here, we are looking at a stacked histogram of the age distribution of these events. The x-axis is time and the y-axis is density, excuse me. And then each color refers to one genome duplication event. Our statistical analysis strongly support a bimodal distribution in terms of the timing of these events, with the majority, the 19 whole genome duplications clustered at 54 million years ago, which is right around the boundary of the Paleocene-Eocene transition. And then a natural follow-up question to ask would be, what happened then? To answer this question, we overlaid a Zachos curve, which depicts the temperature change within the past 60 million years or so. And then we identify that the older peak overlaps with the two super greenhouse events at the Paleocene-Eocene transition. So first of all, we identify a novel geological time period that has been associated with the clustering of whole genome duplication events. During this period, the global temperature has increased by from 5 to 10 degrees Celsius. And then these increase in temperature exhibit a major challenge for tropical lineages like Malpighiales, which has very, very tight thermal tolerance. what might explain the link between global warming and whole genome duplication event is our next question. So one hypothesis, which is termed as a neutral scenario, suggests that the environmental upheaval itself induces polyploids. Here, external stimuli and temperature especially has a pronounced effect on abnormal gamete formation. In rose, experimental evidence has shown that when treated under heat, diploid pollen can form, which is shown by these exceptionally large dyads and triads pollen. And then these can lead to the formation of polyploid individuals in offspring. The other non-mutually exclusive scenario suggests that the environmental upheaval can filter for polyploids because of their higher adaptability. And the evidence comes from a survey in Arctic region that there is an overabundance of polyploids reported in this region. And then the trends is more obviously as you go north. So to conclude for this section, first of all, we identified that the phylogenetic placement of up to 24 whole genome duplications in Mapigillis, and then these events clustered during the Eocene thermal maximum. However, can these whole genome duplication events explain the recalcitrant relationship we see in Mapigillis? Let's revisit the phylogeny again. So here is a phylogeny where we plot these whole genome duplication events to the phylogeny. We can see here all of these events, they are more or less placed on the terminal branches. So theoretically, these events cannot confound the backbone phylogeny. So it seems that, at least for now, whole genome duplication cannot explain the recalcitrant Mapigillis. So in the next section, I will talk about incomplete lineage sorting as well as ancient gene flow. To recap, the problem we dealt with here is really the short internal branches from the rapid radiation. And there are two major factors that has been frequently invoked as a phenomenon to confound accurate species tree estimation. One is high gene tree estimation error. It happens because few mutations are accumulated during these radiative evolution. And then the other one would be the incomplete lineage sorting. So during these rapid radiation, the population size is usually large, so that the gene tree variation can lead to species tree, gene tree discordance as well. Luckily, advances in sequencing as well as phylogenetic inference method has provided new solutions to resolve rapid radiation. For example, the next generation sequencing provide thousands or hundreds of thousands of loci to look at. Which can partly alleviate the lack of information problem here. And then secondly, species tree estimation method that incorporate the coalescence process can accurately infer species tree despite high levels of incomplete lineage sorting. To take advantage of this method, we collaborate with the Lemons Lab at Florida State University. So we compile a phylogenomic data set of more than 400 loci across 64 species that represents all major families in Melpigades. And then to construct the phylogeny, we use both the coalescent as well as the concatenation method. And here's what we got. We have quite a good resolutions towards the shallow level phylogeny. However, for the backbone, many of the nodes, as you can see here, is not resolved above 50% bootstrap support, despite the tremendous amount of data. So what might explain this lack of resolution despite using next generation sequencing, as well as models accommodating for the coalescent process? So the first hypothesis we have is that this lack of resolution can be adequately explained by incomplete lineage sorting, as well as gene tree estimation error. And in other words, the question or the problem we have is a lack of data. To test this hypothesis, we simulate gene trees and then alignments based on the parameters we estimated from our empirical data. And then surprisingly, both the coalescent and the concatenation method can recover the true species tree. In addition, the species tree estimation discordance is much lower in the simulated data compared to the empirical data. So it seems that the lack of resolution need to be explained by factors beyond incomplete lineage sorting. In other words, this is a model violation problem. So to test for the fitness of multi-species coalescent model to our data, we examine the triplet frequency distribution. A triplet refers to a phylogeny that includes 3 species. Under the coalescent model, you will have 2 less frequently occurring triplet whose topology differs from the species tree, which is shown here in blue and yellow. And then due to the randomness of lineage sorting, we would expect the blue tree and the yellow tree to occur at relatively similar frequencies. However, when there is reticulation or gene flow evolved, we would expect one of the minor triplets to occur at higher frequency. For example, the gene flow between B and C would make the yellow triplet to occur at higher frequency compared to the blue triplet. So using this method, we examined more than 40,000 triplets across our phylogeny, and we found that about 500 of them, which account for 1.2% of the total triplet examined, are strongly statistically unbalanced, meaning that these triplets, they violate the assumptions of multi-species coalescent model. And when we map these triplets back to our phylogeny, we found something that makes quite a lot of sense. So first, here, the warmer color means higher proportion of these unbalanced triplets, suggesting hot spots for introgression. And then when we compare this to a plastid phylogeny, we found that a single large clade represents the hotspot for introgression recognized by our method. And then this is also a hotspot for plastid nuclear phylogenetic discordance, because these monophyletic group is recovered as 6 well-supported paraphyletic lineages anaplastic phylogeny generated by one of our previous grad students. On top of that, if you still remember this tangled network of the plant tree of life, the top 3 most unstable nodes are indeed members of these large clade that highly involved in introgression. So it seems that gene flow or introgression can invoke massive gene tree variation that hampers species tree evolution. So to conclude, for the second part, first, Malpighides will still remain one of the most recalcitrant clades among the angiosperm. And then we identify that introgression, which is a previously neglected factor, along with incomplete lineage sorting and estimation error, all of these contribute to the lack of resolution in Malpighides. Finally, in the third part of my talk, I will introduce you to what we found using horizontal gene transfer in a very exciting plant clade, Rafflesiaceae. Rafflesiaceae is a really iconic and fascinating parasitic plant within Malpighides. It is known for producing the largest flower in the world. And it is even more striking that it almost contains no vegetative organs. When it flowers, it will release a flower odor that attracts carrion flies for pollination. There are around 3 genera and 22 species within the family. All of them are specialized parasite of a tropical liana called Tetrastigma in the grapevine family. And then these species live in the rainforest of Southeast Asia. Genomic investigations of these non-model organisms has only become available recently. So to take advantage of that, we decided to sequence one of the parasitic species called Sapria himalayana. We choose it because it has a manageable genome size that's comparable to human. However, its genome is quite repetitive. There are more than 90% of the genome consists of repetitive elements. So to facilitate genome sequencing and assembly, we choose to use a combination of 10x proimmune technology as well as the nanopore technology. And then I received numerous help from our from our staff from the Bauercore facility, as well as the Harvard Informatics group when I did my thesis at Harvard. So a special shout out to these great people. Genomic investigations of Sapria revealed almost immediately some quite unusual features about this plant. So first of all, despite having a big genome, there are quite few genes left, even for the most conserved plant genes. Here on the left, again, is a simplified phylogeny of flowering plants. And then on the right, the yellow bars represent missing plant buscles, which are benchmarking single copy orthologs that used to QC the genome assembly. And then in free living plants, the proportion of missing buscles is quite small. However, when we compared among 3 published parasitic plant genomes, we can find that all of them exhibit an unusually high level of missing plant buscles compared to their close relatives. In addition to losing genes over time, parasitic plants are also known to acquire alien genetic materials from their host via a phenomenon called horizontal gene transfer. It is hypothesized that this process is facilitated by the intimate physical connection between the host and the parasite. So here, the host and parasite is connected by a special tissue called hostarium, which is some researchers thought these are modified roots of the parasite. And then DNAs and RNAs are constantly transferred between the host and the parasite in the hostarium. So this might be potentially one of the molecular mechanisms of how alien genetic materials get fixed in the genome of the parasite. But so far, a lot of work still needs to be done to fully understand how it happened. Phylogeny, again, has been the gold standard to identify horizontal gene transfer. So for a vertically inherited gene, we would expect the parasite to cluster with its close, free-living close relatives. However, if this gene is horizontally transferred from the host, we would expect the parasite to cluster with the host. And here in the Rafflesiaceae tetrastigma system, because the parasite and host are well separated by from each other by more than 100 million years of evolution, this signal is quite easy to be picked up. And this phenomenon, horizontal gene transfer, it has been quite well characterized in Rafflesiaceae already. But one remaining intriguing question is that the age of the parasite is much older than the host. So the question is, there might be some unknown post-parasite association that took place in the ancestors of Rafflesiaceae. Identifying and understanding extinct host-parasite relationship is quite challenging, especially when there's no fossil record. But here, the horizontal gene transfer can serve as a DNA fossil to help us resolve these former host associations. Assuming that a parasite has been associated with 3 different hosts in the past, because DNAs are constantly transferred from the host to the parasite, we can sequence the genome of the parasite and then identify these host-derived gene remnants. Phylogenetic reconstruction of these horizontally transferred genetic material would then reveal the phylogenetic affinity of these host lineages. So to further investigate these phenomena, we expanded our taxon sampling in both the host lineages within the grapevine family, as well as in the parasitic family. So now we have representative species at genus level for both of these lineages. What we found is that, first of all of the younger gene transfers can be traced back to the modern host tetrastigma. However, the older gene transfers have a much more diverse donor lineages. Specifically, we identified here the genus Ampelopsis within the plant family Vitaceae that has been frequently invoked as a donor for these ancient shared horizontal gene transfer, suggesting that Ampelopsis or its close relatives might be the host for the ancestor of Rafflesiaceae. And then the time we are talking about here is probably sometime in the mid-Cretaceous, and then or Paleocene, Eocene. Moreover, we also find that some horizontally transferred genes might serve really important functions. Here, TIC, for example, is an essential gene for pyrimidine and then vitamin B1 biosynthesis. We found this gene to be repetitively transferred from the host to the ancestor of roughly CAC here, as well as more recently to Sapria alone. And given the fact that the native copy of this gene is lost in roughly CAC, it is possible that some of these horizontally transferred genes are used by the parasite as an extended gene pool to supplement genes that has been lost during the evolution of parasitism. So to sum up for this part, first, we use the ancestral horizontal gene transfer to review a history of extinct host-parasite association. And then also some of the horizontally transferred genes are quite functionally important. And then something we haven't fully understand in roughly CAC is that in, for example, nuclear genes, horizontal gene transfer is significant, but only about 1.2% of the genes are acquired from host. However, the landscape changes completely in the mitochondrial genomes. We found that up to 90% of the mitochondrial genes and intergenic regions are from the host. So here is unpublished result of what we know so far about the mitochondrial genome in roughly CAC. It has a quite complicated mitochondrial genome structure consisting of multiple mini chromosomes and maybe linear branched structure as well. And then these blue bars are where we think where we think these horizontally transferred materials reside in the genome. And then these findings actually inspired me to further pursue and inspired my proposed research here at UT, which is to refine the evolution of plant mitochondria in parasitic plants. To give you some background information on plant mitochondrial genome, first of all, the gene content and sequence of these genes in mitochondria are quite conserved across plants. So here we look at cycad, and then from cycad to representative species in flowering plants. You know, these core, especially for these core genes, they are all present. However, there's a study published in 2015 identified a single parasitic mistletoe that's missing a lot of the essential mitochondrial genes. It might make sense because many of these parasites, they acquire their nutrients from the host, and then mitochondria as a powerhouse of the cell, that part of the function might be compromised in this parasite and they could tolerate it. But the question is it a widespread phenomenon across parasitic plants? And then secondly, many of the mitochondrial genes and then they are, I should say, many of the mitochondrial protein complexes, they are encoded by both nuclear and mitochondrial genes. And then one thing we know about horizontal gene transfer is that it introduced alien genetic materials into the genome. And then these mitochondrial complexes shown here in blue, they reflect a very delicately coordinated coevolution over millions of years. And then once some, alien genetic materials is introduced here, do we observe coevolution in the nuclear genome as well to match with what we have in the mitochondria? And then to answer this question, I will propose to use the parasitic plant lineage or Bancaceae as an example. The beauty of these plant family is that it spans the entire spectrum of plant parasitism. You have the basal lineage, Lindenbergia, as a free living species, and then 3 independent evolution of holoparasitism, which means these parasitic plants, they have lost photosynthetic ability as well and completely depends on their host. And then you also have close relatives of these holoparasites that can still do photosynthesis to various level. And then using these plant family, it provides a really nice comparative framework to investigate these following questions. So first, does higher level of parasitism leads to more frequent horizontal gene transfer? And then also, do we observe simultaneously horizontally transferred genes in mitochondrial complexes that's encoded by both mitochondrial and nuclear genomes? And then also thirdly, how does horizontally transferred genes affect function of mitochondria? And here I would receive advice from both Bob and Justin. I look forward to take advantage of their expertise in these areas. And then also, as well as the tremendous resources we have at the Billy Turner Plant Resources Center, I will definitely take advantage of the diversity of our Bancaceae here we have at Texas. For example, if we go to the Big Bend area in the alpine regions, we can find Canophilus. I believe the common name is a cancer root. And then around Austin, you might notice the Castellia, which is called Indian paintbrush, as well as the Aglinus, which is a hemiparasite plant. And then I would end my talk by showing you photographs that I took at the herbarium. For example, here we have a artistically arranged specimen from Oak Ames purchased by Londo. And then this is a specimen of our Bancaceae, the broom rape species that we can find around here in Texas. And then some of the specimens beautifully conserve both the host and then the parasite, as well as the hostorium connecting these two plants. And then herbarium specimens, again, it provides a snapshot of nature itself. What's special about these specimen is that this is specimen of the Indian paintbrush, which itself is a parasitic plant. But on these plants, we find this string-like thing. This is actually another parasitic plant in the Convolvulaceae family. This is the daughter. So here we are actually having a hyper parasitic over a parasitic. I think this is something that's really remarkable that we can find and record by a specimen. And with that, I would like to thank my thesis committee members as well, my co-authors. I want to acknowledge the Department of Integrative Biology for giving me the opportunity to continue my postdoctoral research here. and then also the Stengl-Wyer endowment that allow me to pursue what I want, as well as multiple institutes that has found and support my research. Thank you so much. And I can take questions if you have. Thank you. Okay, let's see. I want to take a question, but I cannot easily see hand raised. So,
Yeah, Ulrich Miller has his hand raised, so why don't you have him first, and then Larry Gilbert next.
So this was really interesting. Thanks very much. That was really well explained. I have a question about part 1 and 2, and a second question about part 3. The first one question is, I think you said quickly when you described the method, said all your data, the information was transcriptomes. Is that right?
for part 1,
Or for
whole
part 1?
genome. Yes.
I was wondering if you had whole genome information, would you get different answers? Like, for example, you refer to the recalcitrance of the older diversifications. Is that because you had insufficient information derived from the transcriptomes? If you had more information from whole genomes, maybe you would get to a different answer?
That's a very good question. So in addition, let me see. We do have genome included in the data set. And then, so here, the blue points are the genomes. And then the beauty of having a genome is that it allows you to verify some of the genome duplications we identified using the reconciliation method. And then you can also visualize the synteny of those duplicated parts if you have a genome. And that's exactly what we did. And then luckily, what we have here is that when we verify our method, our results against the genome data, we get almost exactly the same answer. And then I would say the use of transcriptome may lead to some false negatives, meaning that there was a genome duplication, but not identified because of the missing data in transcriptomes. But we are confident that this method would not give us any false positive answers. Does that answer your question?
Yeah, excellent. Second question is about this interesting finding from part 3 that 1%, about 1% of the nuclear genes were transferred from host to parasite, I think, right? But 90% of the mitochondrial genes. What is the underlying mechanism that these 90% of the genes were transferred one by one, or entire mitochondria were transferred, like the mitochondria were essentially captured by the parasite?
It's an excellent question. And then to be honest, I don't have an answer for a question. I think what might be worthwhile to do is that, again, to use phylogeny to reconstruct a phylogeny of those mitochondrial genes and to see if there is, for example, divergences between multiple copies of the same gene, then to identify if it happens all at once or it happens multiple times. And then the difference is that if that happened multiple times, we would expect some level of divergences or a different pattern of divergence between different.
Maybe the gene order could be relevant here. If there's entire mitochondrial capture, the gene order should be very similar, I would predict. While if it's like one gene by the other, the gene order could potentially be very different. And I don't know how conserved gene order is in mitochondria.
Yeah, and also over this millions of years of evolution, it's hard, but it's an interesting question. Yeah, sorry, I don't have a definite answer for that.
Well, thank you.
Larry?
This was fantastic. I really enjoyed it. And I'm looking at Agellinus and Ampeloxus every day out at our field station.
Sure.
And if the deer haven't eaten it.
That's great.
So this is more of a specific maybe a bit more specific than Ulrich's questions maybe, but with respect to introgression and that aspect of the story, if you look at something like the salicoid clade, you have some very contrasting plants like salix and populus, which are wind pollinated, I assume, and so forth. And then you have the passifloras, which have very specific pollination syndromes and self-incompatibility typically. And it seems to me those would give you very different patterns of introgression, because I would think the wind pollinated ones would be more likely to have some kind of barriers that are, you can put all the other pollen on there and they're just not going to resist it. Whereas in a specific pollination system, you could get a screw up where this bee goes to the wrong flower and then they have no barriers against a hybridization event. And so you might expect that to be more prominent in the insect pollinated kind of lineages. So that, I don't know if you've thought about that, but that would be an interesting comparison.
Yes, definitely. I could definitely see your point. And then here, what I want to say is that the introgression we identified here in Mappikiris is potentially a more ancient one. So if we again look at this figure, the hotspot, the warmest color actually occurs at the common ancestor of a lot of lineages. You just mentioned the Salicaceae here, the Passifloraceae here. So first of all, it's quite difficult to imagine or picture the pollination mechanisms of ancestors at that age.
Yes.
And then going back to your question, I think it definitely makes sense that plants with different reproductive strategies, they would be subject to introgression at different level. I'm not sure there are actually studies look at these or not, but that would be something interesting to explore in the future.
Like I say, it is a more specific or much more lower level thing than you're working with.
Yeah.
Yeah, very interesting though. Thank you.
Yeah, sure.
David Hillis has his hand up as well.
Okay, this is a question about how much we should be worried about radiations that happened a long time ago in terms of getting the exact branching order. And so it's not meant to be, it's really just kind of a thought question. I'm just interested in your opinion on it. So as you pointed out, you can have some lineages that radiated in a very short period of time. And so you might have a bunch of lineages that are lived independently. We know lived independently for say 100 million years. And then sometime about 100 million years ago, they all radiated within say 1 million years of each other. And so the question is, I mean, the reason we're interested in usually in reconstructing the tree of life is because those internodes are often informative and predictive about relationships, not just about relationships, but about the biology. So they can be predictive about common ancestors that two of those lineages may have held, so things that happened at that time. But if the node is short enough, if it's a tiny enough node and it happened long enough ago, it becomes almost inconsequential. And I wonder if maybe there are nodes on the tree of life that we should just not worry too much about when we get down to the level where the reconstruction error and incomplete lineage sorting are just going to be too much of a problem to try to reconstruct them. Those are also the nodes that are likely to be least informative and least predictive about anything, even if we did know the detailed history. So my question is, do you agree with that perspective, or do you think we should really continue to worry about reconstructing very close relationships among lineages that split over a short period of time, a long time ago?
It's a tough question. So first of all, I would think that the bifurcating phylogeny that we used for species tree may seem a little limited based on what we've been discovering recently. There's a so many ancient introgression that have been characterized in animals and plants, etc. So it's more like a network tree of life rather than a bifurcating tree of life. And then I also agree with you that I think some of the nodes, especially the ancient nodes, they are almost impossible to be resolved. This is on my personal experience. By working in the Malpighiales, the short internal branches plus the long external branches. So here we are sort of tripped in a two dark zones. One is the anomaly zone, which is caused by incomplete lineage sorting, too much gene tree variation, and the other one would be the long branch attraction, which in which there's a lot of accumulated mutation that leads to a lot of gene tree reconstruction errors. So I think there is a zone, there is a shape in species tree phylogeny that we cannot resolve even with infinite amount of data.
And so I guess my question was, should we really worry about that? Because those relationships tend to be pretty insignificant, right? It's like, if these things have all been independently evolving for that long ago and they separated over such a short time, those relationships, even if we knew them, would not likely be very helpful or predictive. And so I guess my point is just that maybe there are nodes on the tree of life where we should just all agree. It's like, okay, well, it's just pretty much a polytomy at this point. And they radiated, and we agree they probably radiated in some order in that period of time, but it was so fast and such a long time ago, it really doesn't make much difference. Some people are comfortable with that and some people are not. Some people are just like very insistent that we need to resolve every single node in the tree of life. And I guess my question is this broader one. Maybe there's just nodes on the tree of life where we should just say, okay, well, all these lineages are clearly related, but we don't really know, and it doesn't really matter because they all radiated about the same time.
Well, I'm a bit hesitated to see that. I think there's still might be something that we can do, because the relationship, it does matter oftentimes. Depends on what your question is.
Maybe we should move on to one remaining question. I see Randy has his hand up.
All right, well, first, thanks. It was a really great talk. I enjoyed it immensely.
Thank you.
At least in part because it overlaps heavily with my own interest, but it was a great talk independent of that as well. So this sort of follows on with what David was asking you. I mean, I felt for a long time that this whole notion of assigning, a single evolutionary history to a species or even individuals in some cases is misleading, because you have all this movement of genes from one place to another, and then the problems of lineage sorting and so forth that you talked about. And so I don't know if you looked at your data in this way or not, but I'm curious if you have an estimate, given the large number of sequences that you have for all of your, those lineages, how many different well-supported phylogenies did you actually reconstruct? I mean, I'm sure there's probably a bunch of them that aren't terribly well supported, right? There wasn't enough to give you confidence that it was, something significant. But I'm just curious how many independent evolutionary histories, no matter how they got there, whether it's gene duplication or some sort of lineage sorting or whatever. But I'm just curious how many different histories you were able to reconstruct that you would have confidence in. In other words, how mixed up are all these things? Is there just a relatively small set of histories inside of these lineages or species, or is it really quite a tangle of a lot of different reticulate events?
Okay. I think in terms of the backbone of Malpighiales, I think it would be even difficult to answer your question or give a prediction of whether it's a small or big, because going back to the previous question, we just know so little about what's going on. Then there's a tremendous amount of gene tree estimation error, as well as incomplete lineage sorting and potentially introgression. And then we are not very good at distinguish these three different phenomena apart very well.
Right.
Of course, we can look for signals. They have different patterns in terms of how topology are supposed to distribute. But at this evolutionary level, I'm quite hesitant to conclude on something definite. And then for your question, well, to be honest, I don't think I understand it very well. What you mean by the small versus big? Would you?
Well, so for
reframe it?
example,
Yeah. Mhm.
say you have 2,000 or 3,000 different sequences
Mhm.
that you can analyze, and when you do some sort of analysis of the strength of support for the phylogeny you get back from each of those 2,000 sequences, some subset of them are well supported, and then you can look for congruence amongst that set of well supported ones, and you might find only 3 or 4 of the ones that are well supported. Maybe only 3 or 4 of them actually jump out, or there might be 400 or 500 that are well supported and different. Just as kind of a ballpark sense of whether or not there's evidence that there's a bunch of different histories that need to be sorted out, whether we have the tools for that or not yet, or there's just a relatively small set that seem to be recorded in the genomes.
I see. So the question, in other words, would be, were there highly informative locus that we can find? And then can we only trust these small proportion of locus to reconstruct the species tree? Is that the question?
Well, if you're going to reconstruct the species tree, I wasn't even concerned necessarily with reconstructing the species tree, but just trying to find out the degree to which we can at least potentially discover different evolutionary histories contained within the species, and how many of those are there? Is it a relatively small set with just minor differences, or is it a big set with lots of differences?
I see. So in this case, I would say that for Malpighiates, this specific plant clade, very few, the vast majority of the genes, they are uninformative. And what we can do is that we sort of look at the large group of gene pools and then accommodate all of the background noises, and then trying to parse out signals in those large pool of genes. And the other strategy, the alternative strategy that Randy were talking about here, is to identify genes that is both highly informative and that carries a different evolutionary history themselves.
Mm-hmm.
They are, I think, using some omic methods to screen for these genes that would be pretty hard. For example, would you trust a gene if it just based on high bootstrap support? So it's a tough question. I think one way to approach this might be that if we have some candidate genes, for example, their functions are conserved and we have some candidate, something in mind, that we can specifically look for these genes. But other than that, if we want to screen it or find it from a large pool of genes, I would think that might be challenging.
So I think given the time, a lot of people are leaving, maybe we should thank Li Ming for a wonderful presentation, and she's around. So if you want to talk to her about these things, please come by. Thank you, Li Ming.
Yeah, thank you everybody for having me. Thank you for all your questions. Bye, everyone.
Bye.
Ling,